I'm really happy to speak to you all today. Um, I'm a very visual person, so I'm just gonna do a lot of screenshots and kind of cover and flesh out some of the things that Rob mentioned, which are our main goals. So, what does JBrowse do? It's mainly a genome browser, but it tries to add on a lot more. And there's two specific goals, which are structural variant visualization and comparative genomics. And that's like visualizing synteny and things. And those two things really guide a lot of what you're going to see. And I'll just add a little thing that I think is fun, which is sometimes those are two sides of the same coin. The closer the species are, you're just a structural variant is really just visualizing the comparison between those two things. And we try to support that type of thing. I'll just get into the visual so we can see. So I'm just starting out, a major raw material for all of genomics these days are BAM and CRAM files. And this is showing, our alignments track. It's quite IGV-like, I might say, and we've taken a lot of tips from the IGV team. And so this is actually showing a small deletion that only exists in one haplotype. We can sort and color based on the haplotypes. So we've tried to add a lot of sort of advanced features into your alignments track that do things like that. We also have this little thing I call clipping markers or insertion markers. And it actually sort of counts up how many reads have soft clipping or an insertion at a particular position. And that just gives you a nice quantitative feel for what's going on in these areas. And things like soft clipping, that's where an alignment will abruptly stop at a certain point. So it's a good indicator for structural variance being there. And this is data from the 1,000 Genomes project. Just a similar example with an insertion, and these are PacBio reads that are good at completely spanning the insertion. So it's 1,600 base pairs, but with short reads, you're not able to span that. But we have the show soft clip bases, which is another IGV-ism. It helps you see that there's something going on there. We've also developed these sort of new view types. So this isn't your normal genome browser view anymore, but what it helps you see is a whole genome overview of all of your structural variants in this little circular view, and then a table that lists all of the events. So you're really getting a whole genome picture of what's going on, and you can filter the table on various different columns, or just a simple search bar. You can sort it. And a really cool thing is you can click on any of these little things in the circle. This is a cancer genome sample, so it's very chaotic, but you can inspect any one of these and see the read evidence for one of those events. And so this is the actual data that was used to call those structural variants, and it shows you an interchromosomal translocation, you might call it, where these two chromosomes basically got fused. And so on the top is chromosome 1, on the bottom is chromosome 5, and these long reads were split aligned to those two parts of the genome, which gives good evidence for them being fused. And then the little green bar is just the variant call itself. The black lines are the connections between those split reads. And we call that our breakpoint split view. That's a custom view type, but also our normal alignments track recently added some features that are kind of like this. So if you have paired end reads or split long reads like the previous slide, you can see that on a normal alignments track now, where if two pairs were mapped farther apart than expected, you'll basically get a long arc, and that can indicate that there's a big deletion between that area because pairs should map closer together. And there's multiple different ways of looking at that data. The pink would be shorter than expected, so that would be like an insertion. And the different colors can indicate rearrangements like inversions and tandem duplications. It's a lot to like explain, but I think that like when you're exploring the structural variants, you kind of start getting an intuitive feel for like these types of things. And just this is a toy demo or a toy data example that I synthesized, but I looked at how it might be for like real human data. So this is sort of a large duplication that was called by Sniffles on some PacBio data. And this is what like your normal alignments track might look like. There's a lot of colors. Just a lot of, we're viewing a 200 kilobase region. That's not the largest structural variant you'll see, but it's medium size. And you can't really see what's going on with these, with just the read boxes. But if you flip over to this sort of arc view, you can get a more clear picture that there are things that really flank this coverage increase right there, including from the short reads and the long reads. And I'm definitely really happy with that. The long reads actually look like there's a bunch of spurious arcs in that. I can't really explain that. It might be because the PacBio is a little noisy. It's not hi-fi, but yeah. So that was sort of an overview of structural variant features. And now we can kind of get into some of the features that we're doing for comparative genomics, which I often just refer to as synteny, but there's a little technicality behind there, but I'll just call it synteny visualization. So this is a data example that we like to use a lot. It's this grape versus peach genome. And this is looking at data that was calculated from this MC-scan program. It's actually not MC-scan X, it's called MC-scan Python version. And that just looks at all the orthologous gene pairs essentially. And that works pretty well for like these pretty diverged genomes. But we can also load data from whole genome alignments, which is a different approach. It's not just looking at the gene protein, it's the whole genome. But what's cool with the whole genome alignments is you can actually get base pair exact rendering, I call it, which is like, you can see that if you were just looking at this one, the inversion, you can follow all the little polygons, and that matches up to a deletion down here. So the insertion relative to one is a deletion relative to the other. And that's something you only have with these whole genome alignment approaches. We also have this dot plot view. So that'll tell you, if there's a dot, it matches on chromosome 1 and 18 maybe. And this is a nice way to sort of explore your synteny. You can click and drag on any region and then open from there the linear version of it. So that's sort of this interplay between the two different view types. Oop. Another interesting way you can explore synteny is if most of your genome browsing is just going to be looking at one genome at a time, and you're maybe looking at your gene of interest, and you can open up what we call a synteny track on your plain old linear genome view, right click one of these synteny features, and then say open a synteny view at that position. And what that will do is open up the matching location on the other genome. So in this case, this synteny was just HG19 versus HG38, and it gives you the exact position of that gene on HG38 there. And this red box just means it's like an exact match, because the assembly didn't change that much there. Another thing, this kind of demonstrates something that Rob mentioned in his talk, is that we can show sort of discontiguous regions on the same screen. So this is a gene duplication according to the MC scan data, and it maps to both chromosome 3 and 4. And we can just sort of show those side by side. And if you had, an interest in, what else is going on, you can open additional tracks like promoters or quantitative signals. I just wanted to give a note on scalability here. So currently, we load the entire sort of synteny data set, so like the whole genome alignment, into memory. These files can be really large, especially if you have a large genome or distantly related genomes, because that'll be really fragmented. It increases the file size. So loading whole genome alignments may be difficult if you use a large genome. But also you consider MC scan type approaches, which is just the gene pairs that would match between them. And we're also actively researching ways to overcome these limitations. But I just wanted to mention that because I know people work with large genomes. And I'll just cover a couple other little miscellaneous fun things about JBrowse 2. We added something called multi-bigwigs right into the browser. So this is just a bunch of data from ENCODE, and you can color different groups of tracks as you will. And there's also, this is a multi-row layout, and then you can also overlay them. So this is a tumor versus normal sequencing data set, and you can see, because they're on the same y-axis, that there's CNVs that might be of interest. This is a Hi-C data file. JBrowse 1 didn't have any Hi-C rendering, so all new here. It uses a file format called .hic files, which you might be familiar with from JuiceBox. This one is subtle, but it's often useful for annotators or anyone working on reverse strand features. You can flip around the genome view, and this example doesn't demonstrate, with the alignments wouldn't be that useful, but if you're zoomed in and you're working on a gene, flipping around is very useful. And this is just one more fun one. We got in touch with Nanopore on this, and they said that there's a new standard for drawing methylation directly on Nanopore reads, and so we added the feature to render that, and this is actually showing a lack of methylation. And so we look at every CpG in the genome and say, does the read have a methylation mark here? And count it up with these little red bars. And in this case, there's a lack of methylation there. And also fun, we can create SVG exports. So that can be put into your publications, high quality stuff. And that's that same figure. This goes to our previous question. So we added the so-called URL query param API. So that lets you construct these URLs like, you know, I want to go to chromosome 1, 1000 to 2000 with these tracks open. And there's also similar types of ones. So this is kind of a convenient API explicitly for the linear genome view. There's also one for circular views or dot plot or synteny. That's a little more intricate, but it lets you do it. It does change the URL after you visit this link, but it does, you know, yeah. You can construct links like this, and we think that that'll be really useful for like gene pages, just to like link into the genome browser. And for the admins and model organism database people, you know, we made a CLI tool that helps you load your tracks and your genome assemblies. And we're going to be going over that in that tutorial session following me and following Caroline. But, you know, we just try to make it easy to get these tracks loaded. And for web developers that like to get really into some of the details, we have our linear genome view and our circular genome view as kind of these standalone components, as well as our data parsing libraries if you're making your own tool that just needs to parse some data. And again, we have desktop, runs without a web server. Easy to install. So just for the future, I, you know, always come to PAG and I'm blown away by all the pangenome stuff. I really want to make sure that JBrowse can address that. Currently, the synteny views are very pairwisey, but I want to make it so that we can do multi-way stuff like that. And, you know, write tutorials and how-to videos and just continue with our mission. So that, I really appreciate everyone's attention.
Okay, we have time for a couple of really quick questions. Boy, that was fast.
Just a quick one.
Quick.
Just a quick one. It's about the URL.
Sure.
I saw location, unique IDs.
So yeah, it can't be, so JBrowse 1 let you put like a gene ID in there,
Yeah.
but we don't have that for JBrowse 2 yet.
Yeah.
We could consider adding that though.
Yeah, it could be a yes.
Okay.
Fair. All right. Any other questions? Okay, one more.
Thank you. I think for JBrowse 1, the REST API extensively.
Yeah.
Are there any plans that also for JBrowse 2, or is it already there?
It is not there. We had a little bit of work on that, and hearing your feedback, we'll try to get that pushed through. Yeah.
All right.
What file format is the synteny view use? You want to add that? How do we add a synteny view track?
Yeah, so it's kind of cool. You can actually use the CLI, say add track, and then we support. This is a short list of files. So we have PAF from Minimap2, Delta from Mummer, Chain from UCSC Liftovers. There's a special one called Dot Out from MashMap, and then Dot Anchors from the MC Scan. And that's what we have right now, but if there's others that we should get, do let us know.
All right, we should really move on.
Okay,
Thank you, Colin.
thank you.